Live.
Hello, everyone. Welcome to Workshop 22, Path to a Cure, Targeting CFTR DNA and RNA Repair for Rescue. My name is Daniel Siegwart from the University of Texas Southwestern Medical Center. I will be a moderator today. Joining me is Shingo Suzuki. Shingo, would you like to say hello? Shingo will be a co-moderator for today's session.
Hi, everybody. Hi, everybody. My name is Shingo Suzuki. It doesn't sound working. I'm sorry. Maybe we should go ahead and then start the first presenter.
So today there are multiple ways to target CFTR DNA and RNA for repair and rescue. In this workshop, we are going to hear about ways to use CRISPR-Cas9 gene editing, base editing, nonsense mutation suppression, and antisense oligonucleotide therapies. Whenever you have a question to all of the audience members, please type it into Slido. We will be pulling those questions, and after each presentation, asking the presenters those questions that you submit online. Without further ado, we'd like to begin the first presentation that will be given by Professor Stephen Hart from University College London.
My name's Stephen Hart from UCL Great Ormond Street Institute of Child Health. I'm going to talk to you about our work developing molecular and functional correction of a deep intronic splicing mutation in CFTR by CRISPR-Cas9 gene editing. So I have no disclosures. So CRISPR has emerged in recent years as a potential therapeutic technique by using gene editing. And we're interested in how to harness this method as a therapy for cystic fibrosis. So just briefly to go over what CRISPR involves, it essentially comprises a nuclease, the Cas9 nuclease, and this is targeted to specific sites within the genome by a guide RNA component, which has homology for the targeted region. Having engaged the guide RNA at the homologous target site, which is guided by the PAM sequence, double-strand breaks are made in the genome. Now, in the presence of donor DNA, that double-strand break will undergo repair by homology-directed repair, incorporating the donor into the double-strand break. So this allows for very precise repair of mutations. In the absence of a donor, the other repair pathway is the non-homologous end-joining pathway. This is an error-prone repair methodology that results in the incorporation of deletions or insertions of variable lengths into the target. So it's often used as a very good way to inactivate genes, but not really to repair genes. So in CF, the target cells that we're considering are in the lung, and by methods of inhalation, the cells that would be most readily accessed are the surface epithelial cells. But these cells are post-mitotic and they cannot undergo the homology-directed repair pathway. So we're limited to repair mechanisms that exploit non-homologous end joining. But as I've said, this is an error-prone repair pathway. So which variants are suitable for this kind of repair pathway? And finally, the other question we're working on is how to deliver our therapy in vivo. So the mutation that we've focused on is the CFBE 3849 plus 10KB C-to-T mutation. And briefly, this involves the C-to-T mutation results in the creation of a new cryptic splice site, the 5' splice site at the end of a new intron. And this results in incorporation of a pseudo exon into the mRNA and production of a protein, but this protein is non-functional. So we need to correct this mutation, and the approach we're taking is to target sites flanking the mutation with 2 different guide RNAs that will guide the Cas9 and create a targeted, make 2 double-strand breaks and create a targeted excision, and so restore canonical mRNA splicing. The method of delivery we're looking at is to use the RNP, the ribonucleoprotein complex of Cas9 and the guide RNAs, which can be formulated in a test tube, essentially. We're looking at delivering this with a nanoparticle formulation, similar to one that we've used previously for DNA and RNA delivery. And this comprises a peptide component, which has targeting properties, a targeting domain to target epithelial cell receptors, and also has an oligolysine domain, which combine nucleic acids or proteins by electrostatic interactions. The other component are lipids, DOPE and DOPMA, which are incorporated in the nanoparticle for their potential to disrupt the endosomal membrane. So these particles are formulated in specific order of mixing and ratio of components. And if you get that right, then you can produce nanoparticles of less than 100 nanometers and with a cationic surface charge. So how this will work is the nanoparticle will bind to receptors on the cell surface, be internalized by endocytic pathway, where the lipid components then come into play and mediate escape from the endosome, releasing the RNP into the cytoplasm, which can be guided into the nucleus by the engineered nuclear localization signal sequence within the Cas9. So the first experiment involved the transfection of basal epithelial cells that contain the specific mutation. And we designed guide RNAs and optimized those initially for efficient activity at their specific target sites, and then combine them to create the targeted excision. And so we sequence the targeted region and then perform ICE analysis, which is software for measuring, for calculating the frequency of different indels within the targeted region. And after a single transfection, we had quite a low frequency of our expected deletion event, which you see here. And this is the showing that the preponderance of sites are still wild type, with some single indel events occurring as well. So we then repeated the transfection, which allows an accumulation of editing events. And so you can see that after sequential transfections, we achieved a much greater increase in the targeted excision, which was shown to be somewhere in the region of 60%. So then, having shown a reasonably high level of correction of the gene, the question is, have we restored splicing? So we performed RT-PCR analysis and showed that the mRNA was reduced in size by editing. So here we see the mRNA from the uncorrected cells, and now the mRNA from the corrected cells, and you can see there's a reduction in size in most of the mRNA, indicating successful deletion. This was quantified by fragment length analysis by electropherogram, and you can see by quantification that we increased the wild type transcript to more than 80% in the edited cells. So the next question is, does this successful restoration of splicing lead to an increase in the CFTR protein. So we performed immunostaining in alley cultures. First of all, validated our alley cultures, these air liquid interface cultures for their epithelial properties, and showed MUC5 mucin protein expression, cytokeratin, very characteristic protein of epithelial cells, and also tubulin, which is a protein, acetylated alpha tubulin, a protein within the cilia, indicating the cells are ciliated. And then we looked immunostaining at CFTR and showed substantial increase of the intensity of staining for the CFTR protein. So the protein is there. Now the final question, of course, is that functional? And for that, we perform Usain analysis. It's performed in the laboratory of Professor Baines at St George's University, London. So the alley cultures are placed in chambers within the Usain equipment. And then we add inhibitors and activators and look at their effects on the short circuit current across the epithelium within the alley cultures. First of all, we add a milleride, which blocks the sodium channel within the epithelial cells. And you can see the channel is decreased in activity, and that allows then to look at the increase in activity of CFTR by addition of forskolin, which activates CFTR, although not entirely in a specific way, but nevertheless we see an increase in short circuit current by addition of forskolin. And then to demonstrate that this is specific for CFTR, we add an inhibitor which has much greater specificity, and you see a significant reduction in the short circuit current by this inhibitor in the corrected cells. So this indicates, this combination of activators and blockers indicates that we've restored chloride transport activity in these corrected epithelial cells. On the right here, you see a representative trace of one of the alley cultures showing the activation with forskolin and the inhibition with the inhibitor. So the final question is, can we translate this to an in vivo model? Well, there aren't suitable models in CF mice at the moment that we know of. So we used a model system, the AI9 transgenic mouse, which contains the TD tomato reporter, and upstream of that, there's a stop codon cassette that blocks expression of the TD tomato. This is flanked by LOX-P sites, which can be excised by introduction of Cre mRNA. And we also designed, in an analogous way to the 3849 mutation, guide RNAs that flank the stop codon and that allow for targeted excision. And then we transfected the mouse by oropharyngeal instillation with nanoparticles containing RNPs or Cre mRNA, and showed that in both cases we did see cells where TD tomato expression was restored. By flow cytometry, we showed that in both cases that the quantification of the epithelium was similar, with about 1% of cells corrected. So this shows in proof of concept that we can edit the surface epithelial cells, but the efficiency level is not where we need it to be at the moment, which is probably 20% or more. So that's the focus of our ongoing research. Finally, is the system safe in vivo? Well, we've shown by looking at H&E stained sections of the lung from these mice that there was very little evidence of inflammation. with the RNP, a little bit more with the RNA, but nothing of major concern. So the approaches appear to be safe. So we'll be pursuing this in our ongoing studies. So in conclusion, I've told you a method of using CRISPR and the non-homologous end joining repair pathway to repair a cryptic splice site mutation, and our strategy by targeted excision, the cryptic splice site restored mRNA splicing. We've shown restoration of protein production by immunostaining in Allelic culture models, and that this restored protein also restores anion channel activity. So this looks like a promising approach to developing a therapy for patients containing one or two alleles of this kind of mutation. We've shown that an analogous approach can be tested in an AI9 reporter model, where we restored TD tomato reporter gene expression by targeted excision of the stop codon cassette. And we've described a novel nanoparticle for delivery of the RNPs that seems to work well in vitro and in vivo. Finally, I'd like to thank my colleagues at Institute of Child Health, particularly Amy Walker, who did most of this work, and as well as Professor Baines and Max Woodall at St. George's, and Patrick Harrison in Cork, who designed the gene editing strategy, and Scott Randell at the University of North Carolina for providing cells with the 3849 mutation. Thank you for your attention.
Terrific. Thank you very much for the presentation. To all the audience members, please ask your questions there in Slido, and we'll feed them through to the presenters. Stephen, the first question comes from John Sheridan, and it relates to a question that I had. If you could comment on how were the nanoparticles administered in the animals, and do you have a sense yet of which cell types are transfected?
Yeah, so the nanoparticles were administered by oropharyngeal instillation. So essentially it's a bolus administration, the back of the throat essentially, and the mouse coughs it into the lung. So it kind of self aerosolizes the delivery. It's a method we've used a lot for delivery of mRNA and DNA in the past as well, and we mainly do transfect the surface epithelial cells in the conducting airways, with some transfection of macrophages and alveolar type 2 cells, but predominantly in the conducting airways.
Okay, great. Shingo wants to know, what would be the advantage to cut two sites and make a large deletion instead of deleting either a splicing acceptor or donor? If only one cutting is required, it looks like you do not need repeated dosing.
Yeah, so this is this mutation site is deep within the intron, and there weren't actually convenient PAM sites for doing a single targeted guide RNA. So yeah, that's why we designed the targeted excision approach.
Great. Tom, are you able to ask your question? Go to you next.
Yes. Just a quick question regarding ASOs. I didn't know this alternative splice site looks like it also could be addressed with an ASO. So my question is whether or not that has been attempted, and then how does that relative expression correspond to your technology?
Yes, it has. So correction of this mutation has been reported with other approaches as well, one of which is the ASO. I think that's the group and the company of Batshiva Kerem And that is, I don't know a lot about, I mean, I've seen the presentation, but it works well. I'd argue that maybe the CRISPR approach is going to be more long-term if we're, I mean, it's thought that surface epithelial cells, if that's what we're correcting, will survive for possibly a year or more. So we're not going to need to do frequent administration. There was another approach, CRISPR approach, reported recently, and I think it's been mentioned at this conference, using base editors. So Paul McRae's published using base editors to also correct this mutation. So there's a number of approaches under investigation.
Stephen, for the broader cystic fibrosis audience, would you be able to expand a bit on which patients would be able to benefit from this specific strategy and how you might see this going forward into future trials?
Yeah, so patients that carry this particular mutation, as far as I know, cannot be treated at the moment with modulators. So if they've got one or two alleles, then these patients could benefit from treatment with this approach. As I say, there's a long way to go yet in developing, as with all these therapies, getting the delivery aspect right is going to be a major challenge.
Great. Our next question comes from our friend Patrick Harrison. He writes, have you ever deleted this region from wild-type CFTR to see if there's any negative effects? Asking in the context of a compound heterozygote, as you would delete this region on both the 3849 allele and the other non-3849 allele.
So have we deleted this region in wild-type cells? The answer to that is no. Yeah, interesting suggestion, something we could follow up on.
Great. And then I had one other question. It's related a bit to the translation challenge of many different parts to the CRISPR strategy. Could you comment on whether there are chemical modifications to the single guide RNA, as this has been shown to change stability and plays a role in the ultimate efficacy of any non-viral approach?
Yeah, so these were single guide RNA molecules that we obtained from Synthago. So I don't know offhand what the modifications are, or if that's proprietary information anyway, but that's the source anyway that of the guide RNAs.
Great. Yeah, and I think the last question is from Shingo. He wants to know whether there would be possibility to do repeat administrations in vivo so that the percent of corrected cells could be marched up towards those target numbers that you mentioned.
That's right. Yeah. So I mean that was one of the points. Yeah, that we were trying to demonstrate really that even if single administration doesn't give you the level of editing correction that you need, the fact that these are stable corrections means that as you repeat the dosing, then you'll get an accumulation of effects. And if you're using a non-immunogenic LNP type delivery system, it should be possible to repeat dosing until you achieve the level of correction where you might start expecting to see some therapy. I mean, obviously, what we'd really like to do is have a better delivery system that we can just do it all in one go. But this is perhaps another approach if we can't achieve that level.
Great. Thank you again for the fantastic presentation. We are perfectly on time and ready to move to the second presentation.
Great, thanks.
Thank you. The second presentation will talk about base editing for DNA correction of CFTR, and it's presented by Ms. Sumba Traore. Start the
Hi, I'm Sumba, and I'm from Dr. Paul McCrae's lab at the University of Iowa. And today I'll be talking to you about functional correction of CFTR mutations in human airway epithelial cells using adenine-based editors. I have no relationship to disclose related to this presentation. In CF patients, the lungs are a frequent cause of morbidity and mortality due to respiratory failure and disease. And in the past decades, there have been tremendous advances in the treatment of cystic fibrosis with small molecule modulated therapies that have improved CFTR protein function. However, there still remains about 10% of the CF population that have nonsense mutations or splicing defects that are not amendable with these current small molecule modulated therapies. Therefore, we still find that there is a need to develop new therapies that can repair CFTR mutations for this CF population. There are several methods to target the CFTR gene for correction. One of these methods includes the application of adenine-based editors or ABEs. And of all CFTR mutations, 66% are single-point mutations. And of those single-point mutations, adenine-based editors can repair 46%. Adenine-based editors are a relatively new gene editing tool, and they can change a targeted A to T base pair to a G to C base pair without producing the double-stranded break that is typically associated with the CRISPR-Cas9 system. In addition, it is independent of the cell cycle, meaning that it could potentially precisely edit with very little to no indels in cells that are meiotically quiescent, like those found in the airway epithelia. So I'll just give a brief overview of how adenine-based editing converts a targeted A to T base pair to a G to C base pair. So to begin, you have your adenine nucleotide that is the desired target within your DNA region of interest. Then you have a complex that is composed of a single guide RNA, as well as a catalytically impaired Cas9 nickase that is linked to a deoxyadenosine deaminase. The Cas9 nickase linked with this deoxyadenosine deaminase is what we can recognize or consider as the adenine-based editor. And so when you have this complex, the single guide RNA is responsible for targeting and binding with the DNA locus of interest. And once it binds, it displaces a small bubble of single-stranded genomic DNA. And it's within this bubble where the deoxyadenosine deaminase converts the adenine to an inosine. Following that conversion, the Cas9 nickase recognizes the perispacer adjacent motif, or the PAM sequence, and nicks the strand opposing the edited strand. And it's after this nick that the DNA uses the cellular repair mechanism. And during this repair mechanism, the inosine is treated as a guanine, which ultimately replaces the original A to T base pair with the G to C base pair. So for us, in order to use the adenine-based editors as our gene editing tool, there were a few considerations that we needed to make. The first being is that for any given mutation that we evaluated, we needed to identify if the appropriate NG protospacer adjacent motif or PAM sequence that corresponds with the ABE710 base editor is downstream of the mutation at positions 21 and 22. The PAM sequence is important for the Cas9 nickase to recognize for the nick to occur and preserve the base pair edit. The second consideration is whether the targeted adenine falls within the 4 to 7 base editing window, which is located within the protospacer or the guide RNA. The base editing window falls within that bubble of single-stranded DNA that arises once that the guide RNA binds, and it's also the region where the base editor most efficiently works. After these considerations, we were able to identify cystic fibrosis mutations that can be treated with the ABE710 ribonucleoprotein or RNP. And so we designed short proteospacers or guide RNAs targeting the R5dx and the W22X mutation. So the mutations that we targeted, R5dx and the W12A2X, fall within the class 1 of cystic fibrosis mutations. And these mutations lead to the formation of premature termination codons, which ultimately prevent synthesis of full-length CFTR mRNA and full-length CFTR mRNA and protein. The first mutation that we focus on, that I'll be focusing on, is the R5dx mutation. And so for this mutation, a C converts to a T, creating a stop codon. And so to revert this stop codon, we target the adenine on the opposite strand with the ABE. We designed a guide RNA with the appropriate NG-PAM sequence for the ABE710 base editor. And within this guide RNA, our targeted adenine nucleotide falls within the base editing window, which for ABE710 are positions 4 and 7, which are distal to the PAM. Once the AB RNP is delivered to the cells, the deoxyadenosine deaminase converts the A to T base pair to a G to C base pair, and the catalytically impaired Cas9 nickase nicks the strand opposite to the edit, preserving the base edit and removing that TGA stop codon that we began with. The next premature termination codon mutation we focused on was W12A2X. And for this mutation, there is a G to an A conversion that occurs and creates the premature termination codon or stop codon. And within the guide RNA that we designed, our targeted adenine falls at position 6 in the base editing window. And so after ABE RNP treatment, we should see a conversion of the A nucleotide to a G nucleotide, reverting the stop mutation. And also in this mutation, I have to note that there's an additional A within the base editing window, and this second non-targeted A is known as a bystander edit. So to assess the efficiency of the ABE710NG base editor in correcting the two nonsense mutations, we use the CUFI3 cell line in the primary human airway epithelial cells heterozygous for either the R5dx or the W12A2X mutation. So first we lifted the airway epithelial cells grown at an air-liquid interface. Then we electroporated them with ribonucleoproteins or RNPs, which consists of the ABE710 and their respective guide RNA. After nucleofection, we expanded the cells into a confluency and then placed them back on transwells to differentiate the cells at an air-liquid interface. After about 3 weeks at an air-liquid interface, we ran the cultures through Usain chambers to evaluate electrophysiological restoration of the anion channel, which we were using as a proxy for CFTR protein function. Then we isolated the genomic DNA from the cells and performed next-generation sequencing. These are our Usain chamber results. And the figure on the left is a sample Usain tracing from one of our cultures. The x-axis is the time in minutes, while the y-axis is the short circuit current. We are observing the response to forskolin and IBMX or FNI, which are drugs responsible for opening the CFTR channel. And we're also taking a look at GlyH, which is an inhibitor of the CFTR protein. The blue tracing is a culture from a non-CF donor, and it serves as our positive control, and the red are our mock CF cultures, which serves as our negative control. The green tracing is a culture that was nucleoaffected with ABE710. And so when we open the channel with forskolin and IBMX, we see an increase in current of chloride ions in both the non-CF cells and the adenine-based edited cells. And when we add GlyH, we see a decrease in the current for the non-CF and ABE-treated cells. And the figure on the right is a summary of the Usain tracing we see on the left. And on the x-axis, we have our conditions, and on the y, we have the change of short-circuit current for the R5dx CUFI3 cell line. Each point on this figure is a different culture that was examined, and when we take a look, the mutant mock cells show no response to either FNI or GlyH, while the cells treated with ABE RNP demonstrate changes in anion channel function. From the data, we can conclude that adenine-based editors restore function to the defective anion channel in the R5dx mutation in the CUFI3 cell line. We also examined the primary human airway epithelial cells heterozygous for R553X. And again, we have our conditions on the x-axis, and on the y, we have the change of short circuit current. And we see that adenine-based editors restore functions to the defective anion channel in these cells as well. And the same is true for the W1282X cells and in primary human airway epithelial cells. After Usain chamber analysis, we ran next generation sequencing to quantify the percent conversion from A to G in our cells. And so to complete next generation sequencing, we harvested genomic DNA from the trans wells and used it as a template for a PCR that amplified our DNA locus of interest. We then sent our PCR products or samples to Dr. Newby to further prep and run our samples on an Illumina instrument. And once the run was complete, we used CRISPR-Eso, which is an online tool to analyze the next generation sequencing data. So now we'll just take a look at the NGS data. And so for the R553X mutation using CUFI3 cell line, on the x-axis, we have the mutation, we have the condition, MOC or ABE710 RMP treated cells. And on the y-axis, we have the percentage of total sequencing reads with the targeted A to T base pair being converted to a G to C base pair. For the CUFI3, the targeted adenine nucleotide was converted 91% of the time to a guanine nucleotide. Next, for our primary R553X human airway epithelial cells, the conversion from an A to T to a G to C base pair occurred at a frequency of 77%. And finally, for our W1282X primary HAE cells, we saw a 69% conversion from A to G. Also in this W1282X mutation, there is that non-target adenine within the base editor window or the bystander edit that is converted from an A to G about 16% of the time. So overall, the result that we get from NGS demonstrate that we get high editing efficiency of the targeted A with an adenine-based editor. But because these percentages from NGS involved cells that were heterozygous for the mutation, we had to calculate the allelic editing efficiency to understand the editing efficiency of the base editor on the allele with the targeted mutation. So for the calculation, we took the total percentage of A to G conversion and subtracted it from 50 and then subsequently divided it by 50. For example, the CUFI3 cell line had the percent A to G conversion was 91.1%. And so we subtracted it from 50 and then divided it by 50 to get the efficiency of the base editor on the allele with the R553X mutation. The same was done for the primary R553X and the W1282X human airway epithelial cells, and we see 54.5% and 38% allelic editing efficiency, respectively. In addition, we assessed guide RNA dependent DNA off targets, and we used CRISPR, which is a computational tool online that predicts off targets associated with each mutation. So after entering the relevant information, CRISPR spits out a table of potential off targets, ranking them by guide specificity. And once we obtained these guide RNA off target sequences, we conducted a PCR to amplify each locus of interest for next generation sequencing. So here's our data assessing the guide RNA dependent DNA off target based editing for the top 10 off target sites that we identified through CRISPR. And so for the CUFI3 R553X cell line, you see that we have very little minimal off targets, less than 0.1% off target DNA editing. And the same is true for the primary human airway epithelia cells for heterozygous for the R553X and the W1282X mutation. To conclude, adenine-based editors effectively converts an A to T base pair to a G to C base pair in our human airway epithelial cells heterozygous for R553X or W1282X, ultimately restoring anion channel function. And it does so precisely with very little guide RNA-dependent DNA off targets. So overall, base editors offer a therapeutic strategy to repair CFTR mutations that are not responsive to the current CF therapies. As for future directions, we hope to evaluate strategies for delivery RMP into the cells in a more translational manner. And we hope to do so using amphiphilic peptides or dual AAV Intron-X-Team constructs. We also hope to assess the editing tools in animal models such as mice, pigs, and non-human primates. And lastly, we plan on identifying other CFTR mutations that we can possibly correct with the base editors. I would like to acknowledge my PI, Dr. Paul McCrae, and everyone in the McCrae Lab for their assistance and guidance, especially Satish. And I'd also like to thank everyone at the Broad Institute, David Liu and Greg Luby, as well as members of Belden Therapeutics. Thank you for listening, and I'll take any questions.
Great, thank you for the wonderful presentation. Our first question is, with your NGS analysis of W1282X bystander effect, do you always see the W1282X correction and the bystander, or do you sometimes see W1282 without the bystander?
Yeah, unfortunately, we do see the 1282X when we treat it with ABE710, that it always has that bystander mutation as well occurring in all of our cells.
Do you think there may be ways to design different guides or a different approach to overcome that, or is there always going to be a limitation in W1282X, but potentially not the other mutation sites due to how base editing works?
Yeah, I think with like further improvements on the base editor, we can create better base editors that have a greater specificity for the target mutation of interest without getting that bystander mutation edited, maybe shrinking the bystander editing window, or seeing if there's different PAM sites that we can use to shift that guide to remove that bystander mutation outside of the base editing window. So I think that there are different avenues to do so. Just have to keep going.
That's great. One advantage of base editing likely is that it's not necessary to make double-stranded breaks, which can sometimes increase the precision for gene correction. In contrast, CRISPR-Cas9 may require homology-directed repair, and sometimes they're both NHEJ indels plus correction. In the ALI cultures that you used and presented today, are there different cell types, and is it possible to assess correction in different cell types within the cultures?
Yeah, there are definitely different cell types when we have ciliated cells, basal cells, secretory cells, a number of other different types of cell types in our ALI culture model. As for identifying which cells are being treated, that is something that we still have to work on. And we could do that. I mean, there's a number of people in our lab who use different antibodies and use flow to identify specific cell types within our culture model.
Yeah, I think it's really intriguing because in principle, that may be useful for correction of basal stem cell. So we will all be really keen to understand that data as it emerges.
Yeah, we definitely look forward to doing that, looking at that avenue as well.
Kind of a related question, have you been able to look at undifferentiated and differentiated cells to understand if there is a time or an age of the cell wherein it's undergoing different cell cycle that may or may not be important for base editing correction?
No, we haven't. So no, we haven't looked at undifferentiated cells to see whether or not there's a difference. But it's something of interest. I mean, we have cells growing on plastic that we base edited, but we don't look, so we haven't really done that.
Thanks. The next question comes in. I missed it on the slide. Did you correct the I missed it on the slide. Did you correct the R553X back to an arginine by editing A on the bottom strand or editing A on the top strand to create tryptophan?
Yes, we did. We did correct we reverted the mutation back to an arginine.
Great. So there's a question about the bystander effect, whether you know if that affects CFTR function or not.
That's a great question. We're currently working on seeing if it does. We have a project to see if that bystander edit will affect CFTR protein function, but we don't know how it actually affects the CFTR. There is, in the CFTR2 database, there isn't any reported cases of that bystander mutation causing CF. But in the sick kids database, there is one reported unpublished case that it causes CF. But we're working on a study to really look at that mutation and see how it affects the CFTR protein function.
Great. The next question is, what is the size of the ABE constructs and what vectors are you considering for in vivo delivery?
I'm not sure of the size of the ABEs, the protein. I'm not sure of the protein of the size. It's greater than 160 kilodaltons because that's the Cas9 size for sure, but I'm not sure of the ABE size. For vector delivery, and we have for in vivo delivery, we're working on using amphiphilic peptides. And so in our lab, we've demonstrated that you can deliver protein rapidly and have a transient effect using amphiphilic peptides. And we're also investigating viral vectors, AAV split inteins, to see if we can get the base editor into the cells for correction.
Yeah, I think in principle, one could consider the RNP delivery or a messenger RNA delivery encoding for the ABE.
Yeah.
Okay, terrific. So in the interest of time, we'll move to our third presentation in the session. Thank you again. It was fantastic.
Thank you.
So our third presentation will come from Professor Yi-Tao Yu from the University of Rochester Medical Center.
Okay, I'm Yi-Tao Yu at the University of Rochester. The title of my talk today is Suppression of CFTR nonsense mutations by targeted RNA cellulation. As you all know, cystic fibrosis is caused by mutations in the CFTR gene. About 10% of these mutations are nonsense mutations. Nonsense mutations are the mutations that convert sense codons into nonsense codons or stop codons or premature termination codons, CPTCs, can be any of these three, TAA, TAG, or TGA. So here's the CFTR gene, it's open reading frame, ATG and TAG stop codon here. Normally it is transcribed into messenger RNA, then it's processed into mature messenger RNA, which is then translated into protein according to the open reading frame. The full-length proteins produced. But in some patients, and there's some point mutations that actually change this. For instance, in this codon, G542, there's a single G, the first G of this codon is mutated to a T, then that creates a nonsense codon, a PTC. So this is a nonsense mutation at RNA level, and it's in a GU, and it's a stop codon. And because it's present in a messenger RNA, it triggers a surveillance pathway called nonsense-mediated mRNA decay, or NMD, that degrades most messenger RNA transcripts. Only a small fraction escape in this degradation, and they can be translated, but translation will terminate prematurely at this stop codon, and it stops right there. So in these patients, no full-length protein was produced. So this is the most severe form of disease. So today, I'm going to tell you a new strategy that developed in my lab tackling this problem. And it's guide-RNA guided RNA cellulation to site-specifically target the uridine of premature termination codon, PTC. Okay, so what I mean by that is that we can use the sequence, according to sequence of messenger RNA, design a guide RNA specifically target this uridine to convert this into pseudouridine, which is sufficient to convert this nonsense codon into back to sense codon again. So this way it can block NMD and therefore accumulate messenger RNA and allow ribosome to go through the modified nonsense codon to allow the code continue to go to until it hits the normal stop codon. So we can restore full-length protein this way. Okay, now let me first introduce the Cdu. Cdu is a modified nucleotide derived from uridine through isomerization reaction. This is called the nitrogen carbon bond in uridine, and this bond is first broken, and then this base is lifted up and turned 180 degrees around this axis, and then re-establish 5 carbon, carbon 5, and a C1 carbon linkage, this bond. So this carbon-carbon bond rather than nitrogen-carbon bond. In addition, Cdu has an extra hydrogen bond donor here. So this is sufficient to make nucleotide distinct, okay? The chemical and the physical properties are distinct from that of uridine and any other nucleotides, and it's been extensively studied. Now we know that once in RNA, the Cdu can rigidify the RNA backbone. It can alter RNA structure, it can increase base pair stacking, and also can improve base pairing. And this is a naturally occurring modification, and it is highly abundant and widespread. And then you can find in many different types of RNAs, including tRNA, snRNA, and ribosome RNA, and now also in messenger RNA. Okay, so the question now is how Cdu is introduced into RNA. Actually, there's a major mechanism called Box-HACA guide RNA guided mechanism. So in the cell, there is a large family of Box-HACA guide RNAs, and each family member has a different sequence, but they can all fold into the same structure, hairpin hinge, hairpin tail structure. Each family member is complexed with 4 common core proteins to form an RNA protein complex in a cell. The 4 common core proteins are NHP2, NOP10, GAR1, CBF5, and CBF5 is actually the enzyme that converts uridine into pseudouridine. So during modification, the guide sequence of in the internal loop serves as guides that base pairs with the substrate sequence, specifically positioning this target nucleotide at the base of the upstem. So that is sufficient to tell the enzyme CBF5 which nucleotide uridine to be modified. So this reaction again is a highly specific, and the specificity is determined by the base pair interaction between the guide sequence and the substrate sequence surrounding the targeted nucleotides. And in theory, then, you can take any naturally occurring guide RNA and change only the guide sequence to redirect modification at a different site in different RNAs. And we have done a lot of these experiments and with great success. And today, I'm going to tell you the story about how to apply this technique to suppress CFTR nonsense mutations. Okay, so again, we can, according to the sequence of the messenger RNA, and to design a guide RNA targeting this uridine to convert this into Cdu and then block NMD and allow ribosome to go through to produce to restore the full-length protein production. And here are the experiments that we did. And the first, we took the beta thalassemia gene, or beta globin, essentially beta globin gene. Here there is a PTC at the 39 position. Okay, this is the pre-messenger RNA, messenger RNA, and this is the PTC, UAG, and its surrounding sequences of 30 nucleotides. And we also have a wild-type beta-globin gene with no PTC. And we first did the control experiment and just introduce transfectant, the HeLa HEC293 cells with this wild-type gene, and then measured the messenger RNA level by RT-PCR, or protein level by Western. As you can see, the messenger RNA and the protein are expressed, are present in the cells in a very high amount. Now, we also introduce our transfected cells with PTC containing beta-globin gene or thalassemia gene. Then co-transfected with either control guide RNA or PTC containing guide RNA, or PTC specific guide RNA. Now, in the presence of the control guide RNA, and we see the RNA, the messenger RNA level is really low, and which is not surprising because the NMD degrades the most messenger RNA, a PTC containing messenger RNA. At a protein level, again, we don't detect any protein, which is also expected. But in contrast, when we co-transfect with the PTC specific guide RNA, now we detect in the messenger RNA about 20%, and we also detected the protein, about 10% recovery. Okay, so this is done in the beta-globin gene. How about the CFTR? So we did another experiment simply by changing the beta-globin PTC sequence with a natural occurring CFTR PTC sequence over here. And this is the PTC 542 position, and its surrounding sequences, again, 30 nucleotides long, and complete substitution with this sequence. Okay, now, first, we did the same experiments, and first let's look at the wild type again, and at no PTC there, and we detect the messenger RNA and the protein. But now, if we introduce cells with a PTC containing CFTR PTC containing beta-globin gene, now we even in co-transfected with either control guide RNA or CFTR PTC specific guide RNA, then we measure messenger RNA and protein again. And as you can see in this lane, as the control guide RNA is co-transfected, there is no RNA detected. And the same is true for protein. There is no protein detected. In contrast, when we co-transfect the CFTR specific guide RNA, CFTR PTC specific guide RNA, now we can see detect the messenger RNA and also the protein. Okay, so but here, although we used the CFTR PTC, but it's in the context of the beta-globin gene. So we then looked at a different gene. It's a really a CFTR gene, but it's a truncated the protein gene. It's an N-terminal binding domain. And in this sequence, either it's a wild type or it's a sequence with a PTC at this natural occurring site, G542. Okay, so both a wild type or PTC containing gene. For the wild type, and then we see when transfecting to HeLa HEC293 cells, we detect a messenger RNA and a high level of protein. But now, if we transfect this PTC containing CFTR gene and co-transfect it with a control or PTC specific guide RNA into the cells, then we measured RNA and the protein again. As you can see, the messenger RNA level actually was not changed, and it's just about the same, very small amount reduction. But this is not surprising because the gene has no intron, and it was reported that the NMD is tightly linked to splicing. If you don't have an intron, you don't have splicing, and no splicing, then NMD is not activated. And so even those messenger RNA that contain PTC, they will not be degraded. Okay, that's why the level is similar. But at protein level, you can see when the control guide RNA was co-transfected, there was no protein detected, full-length protein detected. But when you co-transfected with PTC-specific guide RNA, now you see the protein is detected, the full-length protein. Okay, but here again, all experiments described now so far were done in HEC293 cells. What about the CFTR-related cell lines? And we turn to a different cell lines. That is the human bronchial epithelial cells and wild-type cells that contain endogenous CFTR with no PTC. And we measured the messenger RNA and also protein. And as you can see, a fairly large amount of messenger RNA and the protein, two forms, B and C, CFTR, full-length CFTR proteins were detected. Then in the mutant cell line, endogenous CFTR gene has a PTC at this position 542. Now, we transfected the cells with either control guide RNA or PTC specific guide RNA. And then we measured the messenger RNA level and the protein level. And again, in this lane, when we introduced the control guide RNA and no messenger RNA was detected, and the protein was not detected either, full-length protein, no full-length protein detected. But when we transfect the cells with a PTC-specific guide RNA, now we detect the messenger RNA, and we also detect the CFT full-length CFTR protein. Okay, the restoration is about 10%. Okay, now as a conclusion, then I'll come back to this slide again, and this is the mutant and nonsense mutation introduced into this CFTR gene. And as you can see, NMD is activated and the translation stopped here, so you don't have any full-length protein produced. But now we can introduce a guide RNA specifically targeting this uridine and to convert this into pseudo-U and convert this nonsense codon back to sense codon again. That allows messenger RNA to accumulate and then ribosome to go through and until it hits the next stop codon, the normal stop codon, therefore restore the full-length protein production. And finally, acknowledgement. So all the experiments presented today were done by Hiro Itachi, and some lab members also contribute to some extent. And I would like to acknowledge that the experiments were done also in collaboration with Pedro Moraes at the ProCure Therapeutics. And I also would like to acknowledge the funding sources, NIH and the Cystic Fibrosis Foundation. And thank you for listening.
Great, thank you very much. The nonsense mutation remains key on the path to a cure. And I'm excited to hear about this approach using these interesting guide RNAs.
Thanks.
Yital, as you contemplate how this approach could be used as a medicine, could you outline what would be next steps and what would the drug look like and how could that be developed?
I wish I know everything, but yeah, I mean, it's a very early stage. So yeah, next step, definitely wanted to, well, this is just like a test one site, the G542X. And then of course we need to do some other as well. And we wanted to see, of course, this is a point of mutation. If this is not, convert back to the original amino acid, so then the question is whether this is functional. So we have to test for function. So functional assay will be the next, and then and apply to different sites, and then let's see. We can, if it's possible, if we have animal models, then we can go through. So all of those normal steps, but I'm not sure that the timeline and everything. But it's just you just can see that what's going to happen, I think. But of course, in the delivery, there's another hurdle we don't know exactly yet.
Yeah, some of the questions coming through are around delivery, if some modification chemistries could be applied. I have a few questions too, but let me get to the public forum. First question coming in is about the ribosomes. Would ribosomes read pseudouridine as uridine? That's kind of a question.
Okay, so yeah, Cdu is read as uridine and based on pairing, right? I mean, UA pairing, but it's not necessarily encoding. I mean, this is actually, it's very interesting. It's a less, I mean, it's not as specific as uridine adenosine pairing. Sometimes it can really influence not only itself pairing with the opposite, a nucleotide on the other strand, but also influence the pairing on its neighboring base pairing interaction. This actually, I think it is a paper published, this is a collaboration work between me and Venki. This is a ribosome, crystallization, and together, with this sense-antisense codon interaction. And as you can see in that crystal, that pseudo-UA pairing is a lot better than AU pairing. And also, it somehow changes the conformation of its neighboring base. So then, if you look into this, it's a little bit more complex, and the base is flipped out, and makes a very interesting hybrid of Watson-Crick and Hookstein base pair interaction. Yeah, it's a although it's not like changing the identity as I mean, basically it's still in a U, but it can influence the neighboring base pair interaction. That's probably why it rejects first release factor during splicing, so it's not terminates there, and then also allows some sort of tRNA, even if it's not perfectly bounding to that site based on the Watson-Crick rule, but still can bind to that. So that's allowed, that allows it to go through.
The data showed full-length protein. Is a random amino acid inserted, or is there some control over that?
Yeah, it's a very interesting question. We did some, we published, and this is a yeast results, and we know that in yeast, especially, well, I don't know exactly, but at least, in the conditions that we tested, pseudo-UAA or pseudo-UAG codes for serine and threonine, and the pseudo-UGA codes for tyrosine and phenylalanine. But in mammalian cells and under the conditions we tested, we actually checked, found pseudo-U-related GA, pseudo-UGA, actually codes for an arginine. So that is very interesting. So at least in this case, it's an arginine originated, right? The mutations from arginine. So it's very likely we can. Oh, no, not this one. This is a glycine. Yeah, for some of those nonsense mutations can originate from arginine. So in those cases, probably we can make it back to arginine. So that would be great. But still, yeah.
Can you comment on the kinetics of this process? How long can the guide be present in cells? How long will the effect last, and that can connect to a potential therapeutic dosing schedule?
Yeah. So I mean, box HACA, as I said, is a natural RNA, and the modification is a natural mechanism. So the box HACA, once it's in a cell, it's present as a complex, RMP, which is, we know, very stable. So we did actually a time course, And we found we just transfected then at different time points, 3 days, 8 days, and 11 days, 16 days. Then we tested them and we just measured the messenger RNA and protein again. And we found that the virtually not much change except that the cell growth, right? I mean, you have a dilution there. Otherwise, the effect is about the same because we know the RNA is stable. So it should be, I think this is a widely accepted, a concept that the half-life is really long and probably is, can go, live throughout the cell life. So this is, what is currently understood.
Thanks. I think we have time for one last question. There's some questions about in vivo testing. Could this be deployed in an in vivo model that would tell you about the efficacy, but also these conditions of how long the duration of benefit might last?
Yeah, definitely. I think it should, but at this point, we haven't gone that far yet. So yeah, totally.
Great. Well, thank you again for the presentation. We will move on to the fourth presentation in this particular workshop that will be given by Dr. Jenny Kirshner from Case Western Reserve University.
Hello, I am Jenny Kirshner. I am a senior postdoc in the lab of Ann Harris at Case Western Reserve University in Cleveland, Ohio. I would like to thank the organizers of this workshop for inviting me to speak to you today about the rearrangement of airway selective cis-regulatory elements and how this affects CFTR expression and chromatin organization. And I have no relationships to disclose. So a main focus of the Harris lab is in identification and characterization of different cis-regulatory elements in the CFTR locus and trying to understand how these are important for regulating CFTR expression in different cell types. And this is best demonstrated by looking at the open chromatin profiles of CFTR in different cell types, as measured by ATAC-seq, and also by looking at histone modification for an active enhancer, H3K27 acetyl. And I'm showing you these different profiles in both 16-HBE cells here in blue, which is an airway epithelial cell line, and CACO2 cells in green, which are a colon carcinoma cell line. Both these cell lines express CFTR. Their promoter is open in both of them. But you can appreciate that there are dramatically different profiles in the different cell types. So zooming in and looking at some of the upstream regions of CFTR, where we know that there are airway enhancers at -35 kb and -44. In 16-HBE cells, these sites are open and they have the active histone mark, H3K27 acetyl, whereas the site is closed and the histone mark is lacking in CACO2 cells. Conversely, in an intronic region that houses an enhancer in intron 11 of CFTR that is using legacy nomenclature, this is an intestinal enhancer. So we see that the chromatin is open in the CACO2 cells and has the active enhancer mark, but those marks are missing in 16-HBE. And many of these enhancers and different cis-regulatory elements are conserved in similar cell types. So we'll see these enhancers in HBE cells, and we'll see more of these enhancers in human intestinal organoid cells. So what this means for the CFTR locus is that it adopts different 3D conformation based on different cell types, where these different cis-regulatory elements make different interactions with the CFTR promoter, depending on which ones are open and what cells we're looking at. And the way that we can look at this is by performing 4C-seq, which is a chromosome conformation capture technique. And this works by measuring long-range interactions across a region using a specific site as bait. So in this case, we're using the CFTR promoter as bait, so denoted by this dotted red line here. And we can see that in CACO2 cells, which have a lot of the intronic enhancers, that the promoter is interacting more with the intronic region of the gene, whereas in 16-HBE cells, which have the upstream enhancers, we have stronger interactions with that region of the gene between the promoter. Additionally, what 4C-seq has allowed us to observe is that CFTR itself is contained within a cell type independent TAD. So there are these TAD boundaries up and downstream of the locus, which bind CTCF and cohesin, that isolate CFTR from its neighbors. Additionally, we've done a lot of work in deleting different cis-regulatory elements in different cell types and have shown that this has great implications on both looping and its expression of CFTR. So today, what I'm going to talk to you about are two different projects we have in the lab where we've now started to manipulate some of these cis-regulatory elements. In the first case, what happens when we change the spacing between different cis-regulatory elements. And secondly, I'll talk to you about what happens when we move a cis-regulatory element. So first of all, we were curious as to if reducing the distance between CFTR cis-regulatory elements can alter expression, structure, and looping. Specifically, what happens when we alter the distance between minus 20.9 kb cis-regulatory element and the CFTR promoter. So minus 20.9 kb is an enhancer blocking insulator. It binds CTCF, and it's very important for maintaining the normal looping structure of the locus in 16-HBE cells. So when we shorten the distance between the promoter and minus 20.9, we can see by the average of 2 clones that CFTR expression is dramatically reduced. And when we take a look at what's happening with the looping in these deletion cells, we can look from 3 different viewpoints and compare the untargeted cells to the deleted cells and look at the changes in the interaction profiles from these viewpoints. So losses of interactions are shown in gray and gains in interactions in the deletion are shown in blue. So looking at the promoter viewpoint, this red dotted line here, we can see that in the deletion cells, we get losses of interactions with the upstream enhancers in the 5' TAD boundary. Conversely, when we look at the 5' TAD boundary as a viewpoint, we lose the interaction with the promoter. However, we also gain an interactions with these upstream cis enhancers with the 5' TAD boundary upon the 7.1 kb deletion. Also, when we look at the minus 20.9 kb site as a viewpoint, we do see losses of interaction with both the promoter and, gains and interactions with the promoter and with the upstream elements. However, one thing we were starting to notice is that there's actually a great deal of clonal variation in these different clones. And what I had just shown you was the average of 2 clones that had a very strong reduction in CFTR expression. But at the same time, we actually generated a 3rd clone that genetically does have a homozygous deletion, but has an increase in CFTR expression. And we weren't really sure what was going on in these cells until we actually started to look at the interaction profiles. And so what I'm showing you here are the changes in the interaction profiles in these 3 different clones using the minus 20.9 kb viewpoint. Clone 3 is what I had previously just shown you on the last slide. And you can see that clones 1 and clones 3, which are the 2 that have the reduction in CFTR expression, have very similar changes to the interactions within the 2 clones. However, clone 2, which is this clone that has the increase in CFTR expression, has a dramatically different interaction profile than the other 2 clones. And this has now led us to kind of consider and question if this particular deletion is actually disrupting kind of the function of the minus 20.9 kb enhancer blocking insulator. Even though we're not deleting that sequence, we might be impacting the function of this element in these different clones differently. So to answer this, we're currently generating a smaller deletion in this region. So we've moved it away from the minus 20.9 kb element, and we will be assessing these cells to see if these changes and the clonal variation are, if we have as great of the clonal variation when we shorten the deletion, and that's ongoing. Secondly, I would like to talk to you about what happens when we move a cisregulatory element to an ectopic site within the CFTR locus. And specifically, I'm talking about what happens when we move the minus 35 kb enhancer into a site in intron 1. So these cells were first generated by taking advantage of a delta 35 deletion line that we have in the lab. The minus 35 kb deletion line was generated by a current postdoc in the lab, and minus 35 kb, just to remind you, is a strong enhancer in 16-HBE cells. And when this element was deleted in 16-HBE cells, we see a massive reduction in CFTR expression. When we take a look to see what happens to the looping of the locus in these deletion cells, just first looking at wild type cells, using either the CFTR promoter as a viewpoint or minus 20.9 kb as the viewpoint, the promoter again is interacting predominantly with these enhancers and also the 5' TAD boundary, whereas the minus 20.9 kb element interacts very nicely with the 2 TAD boundaries. In the deletion cells, though, we have changes in these interactions. So the promoter is losing interactions with the 5' TAD boundary and the sites around the enhancers, whereas we have increased interactions with minus 20.9 kb and the 3' TAD boundary upon deletion of minus 35. So using these cells now, we have moved the minus 35 kb sequence into intron 1 of the gene. So taking a closer look at this CRISPR-Cas9 homology-directed repair scheme, what we have done is the fragment that we are inserting into intron 1 encompasses the main ATAC peak of open chromatin for minus 35 kb, and it also contains the sequence that has the highest enhancer activity as measured by transient luciferase assays in 16-HBE cells. And we've inserted that here into intron 1, which doesn't have any open chromatin or H3K27 acetyl or RNA Pol II enrichment. So when we generate these cells, again, the parental cell line, the delta 35, loses CFTR expression, but when we insert minus 35 into intron 1, we see a partial restoration of CFTR expression. So this is an average of 2 independent clones. And we see about 17% of wild type expression of CFTR in these cells. Now, taking a look at what happens to the interactions in these cells, again, deleting minus 35 kb leads to a loss in interactions between the minus 20.9 kb enhancer blocking insulator. So this is upon loss of minus 35. And we also see the loss with the promoter. However, when we restore minus 35 kb to the intron 1 site, we can now see that compared to the parental deletion cells, we're gaining a lot of interactions across the CFTR locus. Specifically, minus 20.9 kb is now interacting more strongly again with the 5' and the 3' TAD boundaries, as well as we see minus 20.9 kb interacting with the promoter and also in the region around the insertion site. So just to summarize and go over where we're currently at with these studies is I've shown you that deleting 7.5 kb between the minus 20.9 kb enhancer blocking insulator and the CFTR promoter dramatically alters CFTR expression and looping. And we are currently generating shorter deletions to determine if either the distance from the promoter or the proximity to the minus 20.9 kb element are critical for normal looping and expression. Secondly, I showed you that restoration of the minus 35 kb enhancer to an ectopic location within CFTR partially restores its expression and looping. And we're currently doing studies that will answer questions about what happens to the chromatin signature in these cells. So does chromatin open up at the insertion site? And are histone modifications and RNA Pol2 recruitment altered at all? So together, these studies have provided important insights into important structural considerations to take into account when generating any kind of partial CFTR cDNA insertions for different therapeutic purposes. And I would just like to thank the Harris Lab for their help with this project. Specifically, I would like to acknowledge Manali Nandi Mazumdar, who generated the minus 35 kb deletion line that was used. Two graduate students in the lab, Alec Paranjape and Chi Yin, they're the ones who really do a lot of the bioinformatics work, which this work hinges on. I also would like to just acknowledge our funding. And this is part of a greater collaboration that we have with Brian Davis at UT Health in Houston and members of his team, Christina and Shingo. And thank you for listening.
Great, thank you for that really interesting presentation. I have some questions, but let's start with Slido. Patrick Harrison asks, cool studies. Should us therapeutic gene editors be a bit more wary about big changes we may be introducing, which may dramatically affect CFTR expression?
Yeah, so great question. We also have been doing some collaborative work with some other people who are, putting in super exons and things like that into different parts of the gene. We did this previously with Brian's group with their insert, and their location for their exon or super exons and things didn't affect the kind of known chromatin structure of CFTR in terms of ATAC, so looking at open chromatin, but we haven't really done any 4C-seq studies on those to see if we're changing looping or something like that. But a lot of studies, with intronic insertions, it looks like, if you stay away from the important ones in the cell type that you're going for, you might be okay. But again, we haven't looked at looping in other people's cells yet.
Yeah, I have similar questions if we could stay on this topic for a moment. Current gene editing strategies like the ones we've heard about today, CRISPR-Cas9 and base editing, are initially focusing on known mutations, whether it's the most common deletion at F508del or common nonsense mutations like 542X, G542X, and W1282X. There is a wave of genome editors that are being developed that can insert larger sequences of DNA and potentially the entire CFTR gene at some point. If you don't mind discussing just what your studies reveal with respect to trying to insert or modify CFTR in its natural position versus trying to knock it in at a safe harbor site somewhere else that may not be on chromosome 7. And whether those may be good ideas or bad ideas and consequences that, as Patrick said, us genome editors may not be thinking about.
So, I think some of the studies where, that I just talked about in the talk, specifically the relocation of a cis regulatory element, so we took a very important regulatory element out and put it back in somewhere else in Intron 1. And, again, we went into a region, where Brian's group has also kind of been doing some of the targeting in that general area as well, where it's, nothing much is going on in airway cells. That wouldn't be the case in intestinal cells. So, everything I think is cell type dependent, where you're trying to edit and things like that. The airway is kind of nice in regards to regulatory elements, as the important ones that we know are very important are upstream of the gene. So the thought would be that if you're modifying CFTR in airway cells, intronically, you're not going to be disrupting too much of the normal chromatin regulatory mechanisms there. If you go into some other cell types, so intestine or maybe even pancreas, then there's different considerations to be made, because there are intronic regulatory elements that are active in the intestine and pancreas presumably as well. So it's very specific as to where you're targeting.
I think the next question goes into the topic we're just going through a bit. It comes from Hua Lu. Are there any intron mutations that affect CFTR expression? What about the -35 kb enhancer?
So there aren't really any for -35, I think, in CFTR2. We have done some studies where we've previously gone back and resequenced some older genomic DNAs of patients who at the time only had one known CF allele, and we did find some SNPs and things like that in some of these patients, and did a little bit of follow-up studies with luciferase assays and things like that. But we were limited to having genomic DNA only from those patients, so we were limited in kinds of the studies we could look at with those.
Great. Yeah, it's really fascinating.
Yeah, cis elements, the non-coding region, still really important. Don't forget about it.
Yeah. I'm thinking about the general audience that we have at these meetings. It seems like your studies would have to involve a lot of different kind of methods, from sequencing with wet lab and non-wet lab experiments. And how do those factors play into your daily life?
So I'm pretty much the wet lab person for this. Like I mentioned at the very end, we have some graduate students who have been great doing the bioinformatics work and putting it into a form that, with my very limited capacity to do some coding, I can get some data out of it. But it's really a team effort for most of the work in our group. And even the graduate students who are do the bioinformatics, they also do some excellent wet work as well. But it's a lot of collaborative work, and it's lots of tools in our tool house.
That's awesome. Well, looks like in the interest of time, we will move on to the fifth presentation in today's session. This is last but certainly not least. It is going to be given by William Harris, Professor Harris from UAB in Birmingham.
Hello, I'm Tom Harris. My email is wtharris@uabmc.edu. I welcome any emails after this talk. Thank you. My disclosures are NHLBI and CFF funding. I also have Insight PI for some Vertex phase 3 studies, and Ionis provided the ASOs we'll talk about in today's presentation. So let's kind of briefly talk about the background of today's presentation. So we discovered that TGF-beta, which is a genetic modifier of CF lung disease, regulates CFTR expression, function, and response to CFTR modulators through a microRNA called miR-145. Now, microRNA are small non-coding nucleotide sequences that regulate gene expression. So looking at the mechanism to the left here, TGF-beta increases miR-145 expression. miR-145 then binds to the CFTR messenger RNA, and then it degrades that messenger RNA so there's less transcript available to translate into CFTR protein. Now there's less CFTR protein, so there's less substrate for the CFTR correctors or the CFTR modulators to correct. And this drawing is lumacaptor, which is our previous work. So our interventional strategy is to interrupt miR-145 binding to CFTR Previously, we looked at full antagonism, and in today's talk, we're going to talk about ASO target site blockade. The idea being that if we can inhibit miR-145 binding to CFTR, there's more protein available, there's more substrate available, and so the CFTR correctors are more effective. So we talked briefly about the two bindings, the two miR-145 blocking strategies, one being full antagonism that we've looked at previously, in which the antagonist binds to the miR-145, and miR-145 is not available to then bind its downstream targets. As microRNA have more than one downstream targets, there are some dysregulation of those downstream signals. Instead, we have focused on ASO target site blockade, and in this approach, the antisense oligonucleotide masks the miR-145 binding site on CFTR, so it inhibits the miR-145 binding to CFTR, but leaves the miR-145 available to direct its downstream signals. So there's really three core questions that I'd like to address with this presentation. One is our previous work has again been in the setting of less effective modulators, but now that there is a highly effective modulator, specifically the elexacaftor, tezacaftor, and ivacaftor, which has really transformed CF care, how relevant is this TGF-beta miR-145 access to improve correction? Does TGF-beta still impede ETI correction? Can we still improve ETI correction? We'll address that. Second, how effective and selective is the ASO target site blockade as an enhancer of CFTR correction? Is it as selective as we anticipate theoretically? And third, do nasal organoids provide a human biospecimen that can model an oligotherapeutic response to ASO target site blockade? So the overarching hypothesis is that ASO target site blockade of miR-145 binding selectively increases functional efficacy of CFTR correction. So let's answer that first question first. So how important is TGF-beta and miR-145 to CFTR correction? In this slide, we are going to have the top panel will be ASO, antisense oligonucleotide. So again, that's where we're masking the miR-145 binding site. The bottom panel is the antagonist, where it's directly interacting with miR-145. Then these are functional readouts. So this is forskolin, which activates CFTR channel, and ivacaftor. And then typically four conditions: the control condition, the CFTR modulator condition, TGF-beta, and the ASO restoration. So these are biological replicates, and we can see as a baseline, there's not much CFTR activity. The modulator robustly increases that activity, but TGF-beta continues to squash it, effectively nullifying CFTR correction. And the ASO restores it, nearly completely reversing the TGF-beta inhibition. Now, again, if you look at the cumulative data to the right, it's a little bit of a clearer picture. Control, CFTR correction is robust, TGF inhibition is profound, and the ASO fully restores CFTR correction. We see the similar picture with the antagonist, again emphasizing how important, how potent of an inhibitor TGF-beta is to CFTR correction. A second related question is, even in the absence of exogenous TGF-beta, how much does miR-145 inhibition increase CFTR modulator efficacy? So now we have these highly effective modulators. Can we further increase their benefit? And the answer is yes. So again, we have our control condition, CFTR modulator ETI increases CFTR activity significantly, but then again, on top of that, the ASO increases the CFTR functional activity another 40%. Similarly, the antagonist increases CFTR activity another 50%, and again, that's shown here to the right. So even though ETI is already highly effective, miR-145 inhibition can further increase or enhance CFTR modulator efficacy. The next question is regarding selectivity of the ASO versus the antagonist. And so this is RNA-seq data. The two axes are fold change and adjusted p-value. with a threshold of a fold change greater than or equal to 2, or absolute fold change greater than or equal to 2, and p-adjusted less than 0.05. And what you can see is, as far as the ASO approach, which only masks the miR-145 binding site, we don't see any real perturbation of by RNA-seq. So no transcripts are predicted by RNA-seq to be increased above our threshold. In contrast, the antagonist, RNA-seq identifies with miR-145 antagonism about 400 transcripts that are perturbed above that threshold by miR-145 antagonist. So the ASO approach is as selective as we had anticipated. Another question is it enough to block just the 3' UTR binding site? And so we addressed this by utilizing FRT cells, which are commonly used for CFTR efficacy, CFTR functional assays, but they have the F508del CFTR is just a cDNA without a 3' UTR binding site. And so that can help us understand how important is that 3' UTR binding site to the TGF-beta and miR-145 inhibition activity that we're seeing. And those are the panels to the left, so it's ASO and the antagonist. And what we can see is that in the FRT cells that lack 3' UTR, we see the modulator increase the CFTR activity, but the ASO or TGF-beta or ASO plus TGF-beta has no activity, indicating that's the site of action. A 3' UTR binding site is necessary for miR-145 regulation of CFTR. In contrast, then we added the 3' UTR binding site to the F508del CFTR cDNA. And then what we found with that is that with that addition of the 3' UTR binding site, we were able to replicate what we've seen in primary airway epithelial cells, such that we have the ETI correction, but then we have the ASO boost that further increases CFTR activity, and then the TGF-beta suppression, and then the ASO restoration. So what these graphs show is that 3' UTR is both necessary for miR-145 regulation of CFTR and sufficient that that TGF-beta and miR-145 regulation of CFTR is at the 3' UTR binding site on CFTR messenger RNA. So that's primary airway epithelial cells and FRT cells. Now the next question is, are organoids a model system that we could use to test oligotherapeutic intervention? So is the TGF-beta miR-145 axis preserved in nasal-derived organoids? And the answer is yes. So first, we see that TGF-beta stimulation increases miR-145 expression 4-fold in these organoids. Second, TGF-beta stimulation decreases CFTR expression in half. And then the ASO blockade restores CFTR messenger RNA levels to baseline. And in the absence of exogenous TGF-beta, we see that the ASO further increases the CFTR messenger RNA to increase the substrate available for correction. So now moving from those biochemical measures to functional measures, this is wild-type nasal organoids, or this is establishing the proof of concept that in wild-type cells, that the ASO TGF-beta miR-145 axis is preserved. So these organoids, CFTR function is measured by the swelling of the organoids, which we call that the average fractional change, or AFC. And so what we do, we observe that fractional change over an 8-hour period. These are wild-type cells, so at baseline, which is the black line, we see the swelling. After TGF-beta stimulation, we see that swelling is neutralized, but then the ASO blocks TGF-beta inhibition of CFTR and restores the organoid swelling, and this is quantified to the right. Now, those are wild-type cells. Now let's look at CF organoids. First, looking at miR-145, and does the ASO block miR-145 inhibition of CFTR function? And again, as before, black is control, Now, the ETI, the CFTR modulator, is in green, so we see a response after that. The red is miR-145, and the blue is the ASO blockade. And we again can see that ETI increases CFTR activity, miR-145 decreases it, and the ASO blockade restores CFTR function. Now looking at TGF-beta, we see the same effects. So again, black is baseline, green is CFTR correction, red is TGF-beta inhibition, and blue is ASO restoration. So again, to the right, baseline, minimal function, ETI correction, TGF-beta suppression, and ASO restoration. The last graphs here are answering that question of, does the ASO further increase CFTR function in the absence of exogenous TGF-beta? And the answer again is yes. So even in the setting of highly effective CFTR modulators, ASO or miR-145 antagonism increases CFTR function. So again, we have baseline measure, CFTR correction, and then again the ASO on top of that, as demonstrated to the right, and the antagonist shows the similar effects. So these studies present a couple of findings or conclusions. So first, TGF-beta is a potent inhibitor of ETI efficacy in airway epithelial cells and nasal organoids. And this is a target to improve CFTR correction even further. Number two, both ASO target site blockade and miR-145 antagonism reverse TGF-beta suppression of CFTR correction and increase the efficacy of ETR correction or ETI correction. Three, ASO target site blockade is a selective strategy to improve CFTR correction. So it's as effective as the antagonist, but it's much more selective, and so is a preferred approach. Four, nasal organoids retain this TGF-beta miR-145 CFTR access to provide an accessible human biospecimen to test this oligotherapeutic benefit. And this may be important for low responders to traditional ETI therapy. And then lastly, the utility of ASO target site blockade of miR-145 binding to CFTR is anticipated to extend to additional genotypes and therapeutic strategies, as a 3' UTR binding site for miR-145 exists across mutations. And the underpinning concept is that miR-145 inhibition and ASO target site blockade increases the CFTR substrate that's available for correction. So I'd like to acknowledge the financial support that I've received from the NIH and the Cystic Fibrosis Foundation. Take a moment really to point out Farouk Kabir, who's a postdoc who worked with me, who's done most of this work, Steve Rowe, who's director of our CF Center, and my mentor, the UAB CF Research Center that provides the preclinical models for these studies, other colleagues within my division, and Ionis Pharmaceuticals, who's provided the ASOs for this study. And I welcome any comments at present or sending me an email in the future. Thank you.
Great. Thank you, Tom. As the questions start to come in, I wondered if I could ask you to describe a bit more about the antisense oligonucleotide approach. Presumably, these are single-stranded RNAs that may be chemically modified. Just to help the broader audience understand this approach, could you describe a bit more about what these molecules are and how they can be administered as drugs?
Yeah, so they're, like you say, they're nucleotide sequences that are modified to optimize their delivery and stability, but they really are groundbreaking across disease. And so it used to be there are 3 or 4 relatively rare diseases. There was a real breakthrough with a disease called spinal muscular atrophy, where it helped with the splicing site of that. That was delivered directly to the CNS, and now they're being looked in a wider context. There's always a question with the delivery with a lot of these technologies, but the ASOs have a more feasible delivery mechanism. There's always a question of basically topical delivery or systemic delivery. Certainly, in the context of either nasal or lung, topical delivery is the initial approach. They have been shown in a variety of animal models to be effective. As I indicated in the presentation, Ionis is the company that has been involved in developing this for a variety of diseases, and the CF Foundation has a relatively long-standing interaction with them. They're really one of the leaders in the field. So I think the question again is, so delivery is less of a fundamental question with these ASOs than with some other approaches, although certainly that has to be tested.
Yeah, I think they can be made into drugs, essentially.
Mm-hmm.
Could you comment more upon how they might be utilized in the clinic? Would you imagine aerosol delivery to lungs? Would you try to get ASOs to go everywhere? Would there be on-target and off-target potential effects following intravenous or subcutaneous administration as some ASOs are administered?
Yeah, I think you're right. I think there's a multi-step process. So this is in a lot of ways is a working on the proof of concept. So this presentation was largely looking with the question of, okay, if we already have a very effective modality, can this improve it further? And that's kind of we have ETI, which has been helpful to 90% of the population, but there's two aspects of that. One is, can you actually improve it more? And it seems like we can. And then really the important aspect there is there's going to be this, or there is this segment of the population, maybe 25% that are relatively low responders. And is that a group where maybe one of the regulators that is altered levels of miR-145, which is endogenously expressed in CF, and then similarly, there's a link between that and CFTR modulators. I think initially along those lines would be topical, and I think the first step really, well, obviously, in vivo as far as CF animal models. Then I think we would go nasal as far as seeing how it affects the nasal epithelium. And then I think it would be inhalation. But again, obviously, each one of those steps you want to make sure. Systemic is another direction as far as the delivery and really honing in on the cell types that you want to deliver. So I think the initial approach is more for the respiratory system than elsewise. And then I think, again, after having shown benefit and selectivity for the most effective of the current CFTR modulators, then that really then opens the category of how can this help increase the substrate for other strategies, or if there's a strategy that is effective but that in need of a boost, could this be an application there? So I see it as, again, the ASO itself is not correcting CFTR, but it's just boosting the amount of substrate that's available, and in some ways inhibiting the degradation of CFTR that might be limiting.
The question comes in from Professor Ann Harris. She writes, there is some redundancy in the miRs that target both the 3' end of CFTR and TGF-beta or its receptors. Have you looked at whether, when you block miR-145 binding, you get enhanced recruitment of the other targeting miRs? That might be a problem for miR-145 blockade therapies over time.
Yeah. So the quick answer is we have not looked at that. Dr. Harris brings in a good point that miR-145 is an important signaling modality of TGF-beta, so it's downstream, but there also is a feedback loop where it also has a target on SMAD, which the SMAD proteins are some of the conductors, and so it's quite interrelated. We again have not specifically quantified other microRNAs and how they have increased in the context of miR-145 stimulation.
I think related to that, do you anticipate there could be any advantages or disadvantages to cell type delivery specificity of microRNAs? I'm thinking a bit about some of the work that was done in liver cancer, where delivery of miR-34A to off-target cells led to unexpected consequences. And just getting back to that question Absolutely. Of how it might be combined with modulator therapy and delivered to the right place.
Yeah, and I think that's a really profound. And so we're very excited what we've seen as far as miR-145 blockade, improving CFTR, but miR-145 is really complex, like Dr. Harris says there. And actually, it has a lot of factors. It's probably best established in mechanisms of fibrosis. Certainly, TGF-beta signaling is very broad, and then also has been as far as cellular differentiation. So although miR-145 itself is very appealing, I think those off-target effects are certainly a consideration. This is what makes the ASO approach so appealing. So it's not that we're blocking miR-145 itself, we're just blocking miR-145 binding to CFTR and haven't thus far seen an off-target consequence of that. So the miR-145 is still available to do its other targets. I think you're right as far as the current approach wouldn't necessarily just target a specific cell type, but the approach has the benefit of just targeting miR-145 binding to CFTR. So either in cells that are not expressing a lot of CFTR themselves, or in cells that don't have a lot of miR-145, you wouldn't expect as much off-target consequence.
And the next question comes from Shingo Suzuki. He asks, is TGF-beta miR-145 CFTR axis, is that axis observed in CF patient airways? The activation in vitro without treating is because paracrine from airway cells or in the medium.
Yeah, this is a really profound question. Early on, I really thought it was a consequence when we were seeing the increased TGF-beta and downstream signal. I really thought it was a consequence of just the CF environment, which is, a very inflammatory environment, and seeing that. But again, whether we knock down CFTR or we use, gene-edited cell lines that have not been in that context, we see this increase in TGF-beta and miR-145. Now, I'm not sure whether that's CFTR itself or another consequence of CFTR, such as maybe a hyperinflammatory state in which TGF-beta signaling is activated. But it does seem to be direct between CFTR and between the loss of CFTR in cells outside of the disease context. Great question.
Terrific. Well, it looks like all of the questions have been asked, and that brings us to conclude today's workshop. Today's workshop focused on targeting DNA and RNA. We have seen approaches that include CRISPR-Cas9 editing, base editing, suppression of nonsense mutations at the RNA level, implications for rearrangement and cis-regulatory elements to affect how our field designs insertion strategies in the future, as well as combination therapies of antisense oligonucleotides that can help improve current therapies. I want to thank the audience for joining and posting all of the great questions on Slido. I want to thank all of the speakers who presented their work today. Thank you again. This will conclude today's workshop.